Today we are pleased to have Professor Keith Crandall as our speaker. Going to speak about the multiple omic approach, the computational approach for multiple omic integrated and data analysis. Professor Keith, he's the director of the Computational Biology Institute at George Washington University. So, Professor, at any time you can start, of course, when you're ready. But just before you start, I sent you already a form. So if you have any question, you can submit it at that form. You can also use the chat box here, but well, you can use both of them. And then just remind you that there is actual data by the African Study of Computational Biology and Bioinformatics. And the deadline for submitting a project is on October, so you can submit then if you have any questions space. Otherwise, Professor Keith, anytime you're ready, the floor is yours.
All right, let's see how this goes. It's good to be with you all. I don't know what time it is there. Here it's 10 o'clock in the morning. So.
Well, his is different. It's 3 p.m. here in Morocco, and I think in some country it's 4 p.m.
All right, so hopefully you can see my screen. I've shared the screen here,
yes,
and
yes,
I
yes.
still see all of you.
Yes, we can.
Just arranging my desktop here, there. All right, and we're a small group, so I'm happy to take questions as we go through this. If you're interested, just raise your hand or wave at me and stop. And but I'm happy to take questions, and I certainly won't fill the hour, so have time for questions at the end as well. So whatever you folks are comfortable with, I'm happy to do. All right, so my name is Keith Crandall. I'm director of the Computational Biology Institute at George Washington University. We're confusingly one of 3 Washington universities in the United States. One is Washington University proper, which is actually in Seattle, Washington. One is Washington University in St. Louis. They have to add on the in St. Louis so you don't get it confused with these other ones. And that one is actually where I earned my PhD. And the third one is the George Washington University in Washington, D.C. This is the university that was established by the U.S. Congress to honor George Washington, our founding father. And this is the one that's actually in D.C. So it's, of course, the best of all 3. That's not quite true, but it is the one where I'm currently working. I've worked at 2 out of the 3, so it's kind of fun. Because we're in D.C., we have a lot of interesting research associates, part of whom are at the Smithsonian Institution, which is just about 6 blocks away from our campus. So it's some of the work you'll see that I present is collaboration with the Smithsonian. I'm a research associate there as well. And I'm in the Department of Biostatistics and Bioinformatics. And this is an interesting new department. It was founded in 2019, so we're 2 years old, and our entire existence has been in the COVID era. But we're an offshoot of a former epidemiology and biostatistics department that added a bunch of us from bioinformatics, and then they became a department of epidemiology and now a department of biostatistics and bioinformatics. So we're excited to be there. I also direct the GW Genomics Core to collect a bunch of data. My lab, my group, in the Computational Biology Institute, we do method development. So we like to develop software for us to use in principle, in first and foremost, and then hopefully we can develop it in such a way that other people can use it and do something useful. We have a variety of methods out there. Our most cited method is something called model test for testing models in phylogenetic analyses. This has over 20,000 citations. It's in the top 100 papers in the history of science in terms of citations, which is kind of crazy to think about, but it's because it's just a 2-page application note in bioinformatics, but it's a useful little tool that really does nothing more than is nothing more than a likelihood ratio statistic calculator. But it does so in a phylogenetics context to look at different models, and it works really well. So lots of people use it. And it also exemplifies the way I like to do software development, which is get a great student in the lab, talk about some ideas of a project. The student does such a fantastic job with it that you get a nice publication out of it. And then importantly, I'm not interested particularly in software maintenance. So I love to have projects where the student takes the software and goes away onto their happy life and continues working on the project. So in this case, the student was David Posada, and you can see he's the second author on this next generation model test next generation that was recently published in Molecular Biology and Evolution. And that's because he's done the same thing. So he learned from his advisor that software maintenance is kind of a drag. And so he has Diego Deriba, who's one of his students. So my academic grandchild is now the owner of the model test empire with this latest version. And so David and I have both successfully passed it off. And these 20,000 citations are just the prime, just the original publication of model test. If you look at all the different versions, J model test NG, and things like that, the model test software franchise, if you will, has a huge number of citations. So it's been a pretty impactful software. We have another piece of software called TCS that's almost at 10,000 citations. So if a few of you use that and publish some papers, then we'll be at 10,000 citations. This is a software that does network analysis of gene genealogies. And a couple of newer pieces of software for doing metagenomic and metatranscriptomics sequence analysis. These are the ones I'm going to talk about today. Pathoscope and Telescope. Pathoscope for microbiome research and Telescope for looking at transposable elements, express transposable elements in RNA-seq data. And then, of course, as software developers, we're also, at least a few of us are biologists as well. And we have our own study systems and ask questions that are interesting to us from a range of weird things, from like crayfish vision to human health. And so we apply, of course, our methods, and that's part of the motivation for developing the methods is when we want to do analyses where there are no methods. Or at least no good methods, we develop them. And then we also apply a bunch of other methods. So we have a good, robust concept of what's out there and how well different software work and those sorts of things. So the first piece of software I'd like to kind of clue you in on is something called Pathoscope. We originally developed this software for identifying pathogens in metagenomic data. And the original publication is in Genome Research. And then we did a software upgrade to Pathoscope 2.0 that we published in Microbiome. This is collaborative work with Evan Johnson, who's a colleague at Boston University. He and I were actually both at Brigham Young University before about 10 years ago. Evan moved to Boston University, and 9 years ago, I just celebrated my 9th year at George Washington. I moved to GW. So we've had a long and successful collaboration developing this software. The software is really interesting. It's basically, a lot of people at the time were trying to take next-gen sequencing reads, short reads, assemble them, and with those assemblies try and identify what pathogens looked like. And we kind of scratched our heads and said, you know, it turns out assembly is hard. So let's avoid that. And let's just take these raw reads and map them to reference database and see how well we do at calling species diversity. So we did that. And so the basic concept here is take a bunch of raw sequencing reads, develop a library of pathogens or microbes, which we grab from NCBI. map those reads to this library. And like all good bioinformatics tools, we take advantage of existing useful software. So in this case, it was the useful software was Bowtie 2. So we use Bowtie to map the reads, and then our little secret sauce, our add-on, is a Bayesian mixture model for actually doing the identification, the taxonomic calls of what you believe is mapped from those reads. So our approach is relatively fast. It's highly accurate. And the nice thing about it, unlike a lot of these pieces of software out there for characterizing microbiome, ours accommodates, our method accommodates updating the reference library on the fly. So the reference library has a huge impact on your ability to accurately characterize microbiome diversity. And for a lot of these packages of software, the reference library is hardwired into the software, whereas ours is updatable. I mean, you could literally update it every day if you wanted to. And NCBI is getting new bacterial, viral, fungal genomes every day. So it's useful to be able to grab those updates. So this is the secret sauce. You'll be glad to know this is the only equation of the talk. So, but this is the Bayesian mixture model, and what makes our method better than everything else is this reassignment parameter. So most methods either you have this general problem where you have unique reads or non-unique reads, right? And most methods either take the non-unique reads and throw them away, so you make inferences just on the unique reads, which loses a lot of information, most of the data, or they integrate the non-unique reads with the unique reads, in which case you don't get a lot of good resolution. But what our method does is reassign these non-unique reads to allow them to ride with the unique reads. And that gives us a lot more statistical power than most. So this is the concept. If you see these red sequencing reads, they're non-unique. You have 3 different viruses, and these reads map to all of them. The green reads map to 2 out of the 3, and the purple reads map uniquely to RSV. So what our method does is, in this case, you'd map all these reads just to RSV because they're all consistent with this. And you wouldn't call either of these because they have shared reads. And similarly, if you have some unique reads that map to 2 different taxa in your reference database, and then a bunch of non-unique reads, then you just kind of split those non-unique reads relative to the proportions that you see these other taxa. So you just split those. So you end up not calling things that don't have unique reads, but you end up utilizing all these others in the proportion of these unique reads. And that gives you much better resolution compared to other approaches. So that's kind of the nuts and bolts of our method. Like all good method developers, we're curious whether our method actually works or not, and how it works relative to other methods. So we took some actual data from the Human Oral Microbiome project, and we used some simulation software. In this case, Dan Houston's Metasim. Simulated a bunch of sequencing reads from this raw data using a variety of parameters, which I'll show you in a minute. And then we tried a bunch of different methods that kind of came in different categories. So our method is a reassignment method, like Metamix is another one. There are marker gene approaches, Metaflan, constrains. There are K-mer approaches like Kraken and Centrifuge. Genomic distance approaches, Taxator, and there's a mapping with a maximum likelihood model, Sigma. So we compared all these methods, and we compared them across different read lengths, different sequencing depths, different concepts of sequence dominance. So you know, one particular species is taking up 50% of the reads. So this is kind of a acute infection kind of concept. This is sort of a equal 10 different taxa at at 25% each, 50 taxa at 80%, all reads equal. Different numbers of species. This oral microbiome database had 426 species, so that's the highest number we could get to. So we did 10, 100, and 426, and then looked at all our methods. In total, my graduate student who worked on this, Eduardo Castro Nayer, who's now at the Universidad Andrés Bello in Santiago, Chile, as a bioinformatics faculty there. It's a lot of work, right? 864 different analyses that he ended up looking at. And what you realize when you measure true positives versus false positives, so you want low false positives and you want high true positives. So you want to be up in this neighborhood. And you can see that this is, in fact, Pathoscope as the best method. Kraken is also up here with low false positives, a little higher than Pathoscope and a little lower in terms of true positives, but also up here. Centrifuge is right here near Kraken. They're developed by the same folks and they do roughly the same things. And you can see there are some things like Taxator that are way out here in false positives. And this is a result of using those all the non-unique reads in a kind of a naive way and just throwing them into the analysis. You end up getting, you basically identify, you get a lot of true positives because you're basically calling everything in your database. So you get true positives, but you also get a lot of false positives. So you just basically find everything in your database, regardless of whether it's actually there in your sample or not. A lot of methods here that have very low false positive rates, but not as high true positives, including constraints and Metaflan and Sigma. So on average, they do most of the methods do pretty well in terms of, although, you know, even here, Sigma, you're down to less than 60% true positive rates. So we are happy with this. It kind of showed us that Pathoscope is doing something useful and something seemingly more useful than anything else that's out there. We also, you know, a lot of the community, this is all with metagenomic sequence data. So this is lots of 150 base pair reads from across genomes that are being sequenced. A lot of people are doing in microbiome studies targeted amplicon sequencing. So they're focused on 16S or ITS for fungal studies. So we also looked at performance of 16S itself. And this is actually part of a master's thesis of a student with Evan Johnson at Boston University. And QIIME is the big player for 16S analysis, QIIME 2. This is the dominant software. We compared it, Pathoscope to QIIME and QIIME 2, using Pathoscope with 3 different reference libraries: PathoGreen, Patho whole genomes, green genes is PathoGreen, and then the RefSeq library, just the RefSeq library. And you can see at the phylum level, all 3 methods are pretty good, although the variance is much tighter with Pathoscope compared to QIIME. Again, at the class level, now Pathoscope is doing better, both in accuracy as well as variance, much tighter variance. And then at the ordinal level, much better in accuracy and much better in variance. And it just keeps going as you go down the taxonomic scale. The difference, the spread between the differences between QIIME and Pathoscope become bigger and bigger. So family level, genus level, and then the species level, they're not even comparable. And Pathoscope works better, but not nearly as well with the green genes database as it does with our whole genome library or the RefSeq library from NCBI. So capitalizing on that whole genome data, even with just 16S, gives you much better resolution, especially at the genus and especially the species level with Pathoscope. So it's a very powerful approach. So we're reasonably happy with it. And now I want to show you some of the studies that we've been able to do with this to give you an idea of the insights you can get. So one of our first examples where we used this study was using in a pediatric asthma study at Children's National. So you have a bunch of kids that have asthma. You take a nasal swab, which, you know, I used to have to explain, but after COVID, everybody knows what a nasal swab is now, and some people are more used to it than others. You literally sequence the kid's snot, right? So you get millions of sequencing reads, and then you use our software to map those reads against reference libraries, and then call what you see in those data. In this case, the collaborators at Children's were actually doing RNA-seq. So this is, you can use Pathoscope with either the RNA or DNA. One of the interesting things with microbiome studies when you start doing this is you quickly realize that depending on the sample, you get very different proportions of host to pathogens. So you have to adjust your sequencing depth accordingly. So with the nasal swabs, you get 90% host sequences and 10% pathogen. So the first thing that the Pathoscope does is actually map all those sequencing reads against the human genome, and then partition the human genomic data from everything else. And then we characterize everything else in terms of microbes. So if you're using, for example, stool samples, this looks more like 50-50. If you're using, we've done breast milk. Breast milk is like 99% host and 1% pathogen. So you have to adjust your sequencing depth to get a good pathogen representation. The other powerful thing about Pathoscope is that because it's metagenomic data, you sequence everything that's in there. So you can get fungus, virus, bacteria, everything that's going on. So we're going to focus on this 10%, run it through Pathoscope. Our end results, here we have cases, asthmatics, and controls, and you get some proportion of mapped reads across a wide variety of taxa. And one thing that we noticed in the asthmatics is they seem to have a much higher, statistically significantly higher proportion of something called Moraxella catarrhalis, right? And being good bioinformaticians and bad microbiologists, we didn't know what that was. So, but we know how to use Google. So we did the Google search and looked up Moraxella catarrhalis, and Google said it's a upper respiratory disease bacterial. And we thought, okay. So we called our friends at Children's and said, yeah, it looks like Moraxella catarrhalis is causing these kids asthma in a lot of these cases. And then they said, how do you know it's infective? And we looked at each other and said, we'll get back to you on that. And we had a lab meeting and talked about it. And we realized that we still have 90% of the reads of the RNA-seq reads that are from the host. So we can actually, and because this is RNA-seq data, we can actually investigate the host immune response to an infection by Moraxella catarrhalis using the exact same data. So no more money to sequence more things, just more computational time to do the analysis. So when we did this analysis, we were fortunate in that we found a paper by another group that had identified like 70-some candidate genes associated with immune response to Moraxella infection. So we did the differential gene expression with those RNA-seq data on all these genes that are known to be associated with an immune response to Moraxella. And we found differential gene expression, a whole bunch of them. So we could identify, in fact, that in these asthmatics you had an immune response. And so we called back up our collaborators and said, now we can show that. And they said, okay, now we can publish a paper. Just to warn you that these kinds of metagenomic studies end up with lots of data. So it's good to have a good computer and lots of data storage capacity, because you generate lots of data and you need a place to put it. And not only that, but running these analyses, one of the things that a lot of people don't think about is they worry about the primary data, right? We just sequenced all these patients and we have, all these billions of nucleotides, terabytes of data. But when you start doing these downstream analyses, it turns out they generate a whole bunch of data files too. And they're kind of large too. So we when we first started doing these sorts of things, our high performance computing cluster was not particularly large. And we started getting a lot of nasty emails from our systems admin people asking what we were doing with all these data files. And we assured them we were actually doing research, and that's what the computer was for. So chillax, we'll be done with it in a little bit. And so we've cultivated our relationship with the systems admin people, and now they know who we are and what we're up to, and that we sometimes take a lot of computing resources, but we're pretty good at, we have a different system for data storage, so we're pretty good at moving data on to the HPC, analyzing the data, and then moving it off as quickly as we can, so we don't tax the HPC resources per se for the analyses. And then we also have partnered with the systems admin, our Department of Information Technology, to basically upgrade the HPC to a much more robust machine. The other nice thing you can do is I just showed you how to look at the immune function of the microbes, but you can also do the same thing, well, for the host, but you can also do the same thing for the microbes because you have RNA-seq. So you can start analyzing what's going on in terms of gene expression for the microbes and start looking at different pathway analyses using something like KEGG pathways to look at differentially overexpressed or underexpressed pathways going on. And I'm going to show you some additional tools in a little bit where we augment that even further. So the pathway analyses with the RNA-seq really gets you some interesting and useful information in terms of functional analysis, not only in the microbes, well, not only the host, but also the microbes. So you can really look at what we call dual transcriptomics, looking at what's going on in the microbes and the host in terms of the RNA-seq analysis. So it's a very, very powerful approach. It costs a lot of money, right? The RNA-seq to do metatranscriptomics costs probably twice as much as doing the metagenomics on the data. I've yet to convince anybody to pay for my sort of beautiful data set, my preferred data set, which is getting DNA metagenomics and RNA-seq metatranscriptomics all on the same individuals in a case control setting. So hopefully someday I'll convince somebody to do such a thing so we can look at everything and really look at what's going on, the differences between the DNA that's there plus the RNA, the expressed information in the RNA-seq data. So that's an interesting thing. So we not only do human health sample examples, so that was an example, a collaboration with Children's National, Washington, D.C. But as I said, D.C. is a fun place because there are all kinds of folks here, including the Smithsonian. And they have partnerships in South Africa with Kruger National Park on endangered species studies. One of these endangered species is the black rhino. So we're interested in doing a comparison of black rhino microbiome in wild populations from Kruger compared to captive rhino populations in the National Zoo here in Washington, D.C. and other captive populations around the world. And the problem is that black rhinos have these something called iron overload in captivity that they don't seem to have in the wild. And they hypothesized that it had something to do with their gut and the gut microbiome that leads to obesity. And there's this weird pathway of insulin resistance and iron overload and leads to disease and issues with the black rhino. So we thought, well, we can at least tackle the gut microbiome and see what's going on here. And then in a follow-up study, we're actually now working on some of these issues by adding additional metabolomics and proteomics data to the mix. But I'm going to show you the microbiome data to start with, because that's what we have at the moment. COVID kind of ruined our sampling capacity for the last year and a half, so we haven't made much progress on the on the follow-up study yet, but we're hoping to get back into the field here shortly. And I've been fortunate to have to host a number of Fulbright scholars in my lab from around the world, including various parts of Africa, and one of whom was a postdoc. One individual was a postdoc in my lab, Saville Daniels, who's now at Stellenbosch University on faculty there, a full professor. And he's, we wanted to do the sampling of wild individuals in Kruger, but you can't send fecal samples through the mail into the US because of USDA regulations. So we sent the fecal samples to, we have veterinarians at Kruger that my colleague at the Smithsonian knew. She could sample get the fecal samples at Kruger and then send them to Stellenbosch. And then my former postdoc, Saville, could extract DNA and then send us the DNA for sequencing. So one of the beauties of the black, and then a captive is easy because the beauties of black rhinos, there's lots of material to work with. So all of a sudden my freezer is filled with black rhino fecal material because there's no shortage of it. We had 17 wild rhino fecal samples and 8 captive samples. And this was recently published in Scientific Reports if you want to take a look at that. And the idea was to really compare the captive and the wild, and you can see the wild and the captive are very different in terms of their diversity. We also were looking at a couple of different extraction approaches as part of this project to do additional research. And the upshot with the extraction approaches is we love the Zymo kits. The Zymo DNA RNA later kits are really good and gave us much better results in terms of raw number of sequencing reads, much better material for sequencing. But in terms of the biology, it turns out these wild versus captive guts are very, very different. And as you might imagine, the captive guts basically look like a typical horse gut microbiome here in the US. So in the captive, the wild samples look like something totally different. And this is kind of one of the weird, interesting things about this study that came out was that, when you do a human gut microbiome from a human in the US, you can map 90% of those reads, you know what they are. When we do gut microbiome from black rhinos, half the reads we couldn't map, right? So we still don't know much, frankly, about bacterial diversity, and certainly not much about bacterial diversity in undersampled parts of the world in terms of human microbiome, and almost nothing about organismal microbiome diversity in organisms other than humans. So I think there's a huge opportunity for basic research and basic characterization of microbial diversity, identifying new species out in the wild, sequencing them, providing those genomes for reference databases so we can do a better job at the metagenomics kind of things that capitalize on those. But with the 50% of the reads we could map to something, those somethings are very different in the wild versus the captive population. So and so we basically recommended a wholesale change in the kinds of diet that they're feeding the captive rhinos to see if you can change that underlying gut microbiome diversity. And they're trying this at the National Zoo in Washington, D.C. now. The other interesting the other interesting thing about microbiome research is that it's great for teaching, right? So we actually have developed a project. I have a colleague in the Computational Biology Institute who teaches genomics, and he was doing a teaching workshop, and he said, he wanted to integrate a project throughout the course, and was talking to me about ideas. And I said, why don't you test the grandma hypothesis? And he said, what's the grandma hypothesis? And I said, I basically was making this up. I said, well, this is my grandma, right? My grandma is 1/4 Cherokee, Native American. My grandma, used to take, this is not myself and my two brothers, although there are three of us boys, and she used to take us over the summer. My parents would dump us off for a week or two with grandma and grandpa to spend in rural Indiana, to hang out with them while they went and had fun. And we loved it because we loved hanging out with grandma. She'd take us fishing and do all kinds of fun stuff. So, but one of the things she did was, you know, because we went fishing and things, we'd get filthy dirty because we were three little boys. So she'd stick us in the bathtub and soap up a washcloth and hand it to us and say, now make sure you scrub behind your ears and between your toes and in your belly button, right? So I said to my colleague, you know, let's test the grandma hotspots. Let's have students swab behind their ears, between their toes and in their belly buttons, and ask if those regions are significantly different in diversity compared to control regions of a forearm and the back of the calf. So we have two control regions, three hotspot regions, and we're going to test differences. And the fun thing about this is during the course of a semester, the students can take this and they learn everything, right? Starting with learning how to sample, doing sterile samples, swabbing yourselves or each other, right? And you do 5 different swab sites, developing a lab notebook and writing down your procedures for everything that you do throughout this project, throughout the semester. So we have formative assessments and a summative assessment at the end, but the formative assessment here is developing your lab notebook, writing down all you're doing here. You can take these swabs, extract DNA, run that DNA out on gel for electrophoresis. You learn about DNA extractions. You do PCR amplification of 16S because that's the cheapest thing to do. So we didn't do the metagenomic sequencing. We couldn't get the university to pay for that, but we got them to pay for 16S sequencing, so we went with it. So they learn PCR, they learn gel electrophoresis. They have a formative assessment of looking at that gel and the PCR products. We run them out on the DNA sequencer, so they learn about targeted amplicon sequencing. We walk them through the genomics course so they can see the machines and talk and learn a bit about DNA sequencing technology from the lab director there. And the formative assessment is their data, right? Do you actually get quality data or not? And then most students do. And so we can run those data through Data 2 or whatever your favorite microbiome analysis packages, because this is 16S and Data 2 has a web interface, so Pathoscope does not. So even we abandon our own software when it's appropriate. And Data 2 and Microbiome Analyst both have very nice web portals to analyze data. It's pretty straightforward. Our software is not quite as straightforward for students to use, but these two allow you to get relative abundances quickly and then do some statistical tests. And then at the end of the semester, they write a scientific report. And of course, what we found was really interesting. The arm and the calf had higher diversity than the ear, navel, and toes, in between the toes. So grandma hotspots actually were statistically significantly different compared to our control areas, but they had lower diversity, right? And we argue that this is because there's basically a few dominant species in these not cleaned environments, whereas cleaning regularly allows more diversity of bacteria to infiltrate those areas. So we've developed we have a paper in the Journal of Microbiology and Biology Education that outlines all the activities for this approach, all the educational activities as well, and provides really detailed instruction on how to implement this in your own lab. This was also a fun paper because I got to work on it with my daughter, who is an educator, and she helped us with the educational component of that study. So it was kind of fun working with my daughter on a paper about my grandma, her great grandma, and our experiences with myself and her dad and her uncles in the bathtub at grandma's house. So the next piece of software I want to tell you about is Telescope. This is an offshoot of our Pathoscope software. It uses the same Bayesian reassignment method only now we've modified it a little bit to identify transposable elements. The hallmark of transposable elements are 5' 3' LTR, long terminal repeats, with some coding sequence in the middle. So that's what Telescope looks for. And it kind of adjusts for the same problem, that a lot of times you have unique reads and ambiguous reads, and there are different software that do different things, but our Bayesian reassignment method allows you to assign some of these ambiguous reads to more targeted taxa based on what they're traveling with in terms of the unique reads. So we've implemented this in software. Matthew Bendall was a former PhD student in my lab who's the principal author of the software. He's now at Weill Cornell Medical School with Doug Nixon, who's a former collaborator, a former faculty member at GW as well. Transposable elements make up 45% of the human genome. They were discovered by Barbara McClintock by studying what we call here in the US Indian corn that has this mosaicism that's caused by transposable elements. McClintock discovered that, demonstrated that these transposable elements were the cause of this color variation, and won the Nobel Prize. She's still the only female to win the Nobel Prize unshared in the category of medicine. So it's a huge discovery, and lots of people have been doing mobile element work as a consequence. It turns out that recent studies there are a lot of hot topics these days showing that these expressed transposable elements are associated with diseases, that they cause disease, they disrupt normal function, they are associated with a variety of human genetic diseases. So we're very interested in analyzing them, and our Telescope software provides a new opportunity to really see what's going on. And these transposable elements for me are just another part of the microbiome. And we're specifically interested in microbiome and cancer. And we actually published this review paper a couple of years ago in Trends in Cancer to look at the broad aspects of what the microbiome and what is the microbiome, how is it associated with cancer. And specifically in this study, we're interested in how Telescope can identify these transposable elements that are associated with prostate, breast, and colon cancers. So here we're analyzing RNA-seq data from the Cancer Genome Atlas with a variety of data, with these 3 different data sets with different numbers of individuals. The most robust data set, 111 individuals for breast cancer, 52 in prostate, and 24 colon cancer patients. This is great data because it's tumor normal tissue from the same individual. So the controls are as nice as you want them, as you can get them, by sampling the tumor tissue and then neighboring normal tissue, doing RNA-seq on both those. Again, large volume of data, 70 million reads per sample, except for colon cancer, which is a little less sampled, 56 million reads, but still lots of reads per sample. And we can take these and look at, and there's a lot of good, one of the fun things about this, there's a lot of good phenotypic data like Gleason scores and tumor stage and things like that. So you can start analyzing some interesting aspects of this. When you partition the normal and the tumor, you can see you get normal separated from tumor almost exclusively. This is prostate and breast cancer. When you look at differential gene expression, you get a lot of genes that are differentially underexpressed and differentially overexpressed. And you can do the same for the transposable elements as well. And in this figure, we mapped both the transposable elements. In this case, we're focused on human endogenous retroviruses. So these human endogenous retroviruses, and then their neighboring genes. So here we've used what's called a Circos plot to plot these out in terms of chromosomal location across the human genome. So this is chromosome 1 through 22, X and Y, and then you can see that these are the human endogenous retroviruses and their relative differential expression levels, and then associated genes, target genes in the host genome. So again, the beauty of the RNA-seq data is you can investigate what's going on in the host as well as the gene, as well as the microbiome, in this case, human endogenous retroviruses and figure out what's going on. And there are some intersecting HERVs. Now we're doing deeper studies into some of these human endogenous retroviruses and asking the question, how broadly impactful are they across different cancers? You can see there's huge numbers of these expressed HERVs. There's large numbers of differentially expressed HERVs, and some of them are quite common across some of these different cancers that we've been looking at. So we also have looked at lupus, systemic lupus erythematosus. It's an autoimmune disease where the immune system attacks its own system. It's widespread inflammation, and it impacts the inflammation system and does tissue damage, impacts women mainly of childbearing years, and it's more prominent in the US in folks with African American ancestry. There's no known cause. The implications, some folks have done genetic studies and looked at genetics. People have suggested environment, hormonal factors. We wanted to look at HERVs and HERV expression in particular to see what's going on. There are a lot of overexpressed HERVs this is the same kind of sampling, cases and controls, highly active and inactive lupus. We've looked at a couple of different data sets. These are whole blood cell samples. One found over 13,000 expressed HERVs, 321 differentially expressed HERVs between cases and controls. The other whole blood cell data set found over 12,000 HERVs with 160 differentially expressed. So these are the HERV differential expression. Again, you can look at the differentially expressed HERVs and the differentially expressed genes because it's RNA-seq data. You can look at them in a chromosomal context and see what's going on and see where these are being impacted. And because of these telescope results, and we wanted to do a more formal analysis of looking at which pathways were enriched for these HERV elements and how the HERVs associated with some of the host genes and host pathways. So we've developed, or we're developing another software suite called Omics that has some other tools, some machine learning tools, Maslin, to look for pathway associations, and DeepPath to test for pathway enrichment for the differential gene expression and identify, for example, in this case, response to interferon, to type 1 interferon, response to virus. So there are certain pathways in the host genes that are being differentially expressed as a consequence of these differentially expressed transposable elements. So it becomes a really powerful set of tools that you can use to look at pathways. And is ultimately we're heading towards this concept of multiomics data integration. So taking the metagenomics data, metatranscriptomics data from hosts, from pathogens, from transposable elements, adding to it metabolomics data, proteomics data, and integrating it all for testing for differences and localizing those differences across genomes and associating with broad scale phenotypic differences. So we've done, as you might imagine, a lot of COVID-19 work all of a sudden. And in looking at the COVID genome, we can identify regions of the genome that show associations with different clinical features. And using this deep path analysis software, and our OmClust is another piece of software within this Omics suite for identifying clusters. It's a more powerful approach than doing principal components analyses. But here we're looking at gene regions that seem to have significant clusters of diversity. And then we can associate those gene regions with different phenotypic aspects of COVID-19. So the country of origin, the exposure, the sex, the age, the region. We even identified we developed a new statistic called Coherence. So you can look at how similar or different COVID SARS virus genomics is relative to other genomes in the neighborhood. So you can see in a few of these states in the US, like Indiana, New York, I think that's South Carolina, Louisiana, have higher diversity than in the states than you would expect, whereas others have about as much diversity, and others we don't actually have any that have lower. So this is indicative of basically the higher diversity is indicative of diverse elements coming into the state. So you can use this globally as well. And you can also use our deep path for identifying regions of the genome that are associated with well, here we're analyzing metabolomics data and finding metabolomics pathways that are associated with different disease states. So this stuff is all, the Omics software is under development. We're applying it to a lot of COVID data and to a few other data sets. We just got a new grant to do even more of this development for the next 5 years. So it's very much ongoing work in progress. But it's a very powerful approach to linking, broadly speaking, linking genotypes and phenotypes, but specifically a wide variety of genotypic data from metagenomic data and other kinds of omics data in doing association testing with those in a powerful way. So just to give a bunch of thanks, Evan Johnson's my collaborator at Boston University, working on Pathoscope, Matthew and Doug Nixon at Weill Cornell, working on Telescope. Ali Ranavard is the principal architect of Omics I. He's at our Computational Biology Institute at GW. Kate Cepinelli has worked on cancer and HERVs, collaborates with us with that. Lots of great graduate students working on a variety of these projects. Buddha is at the Smithsonian working on the black rhino stuff. Maggie Steiner, we have got lots of great undergraduates at GW. Maggie was the one doing all the analyses for the cancer study, and we get funding from a variety of places. And then finally, thanks to you, and thanks for the invitation to come talk to you all today. Metagenomics, I think, is really an amazing opportunity for characterizing microbiomes and host taxonomic, taxonomic diversity in the host and microbiome, in the host microbiome, and functional diversity in both the host and the microbes. So you can we've used the metagenomics to look at genomic features of the host and the microbes and transcriptomic features of the host and microbiome. So metagenomics, metatranscriptomics is a really powerful set of data. We've developed a suite of tools now, Pathoscope, Telescope, Omics I, that can take these data and really do some powerful analyses with them. As you all know, informatics is becoming the essential skill even for biologists. So encourage everybody to get some kind of informatics skill sets. Our software is all open source and all available on GitHub, so you can grab it, download it, hack away at it, play with it as you like. And we continue to develop it. As I said, we have a long history of hosting Fulbright scholars and other foreign supported scholars in our lab as postdocs, as visiting graduate students. If there are individuals out there that are interested in coming to Washington, D.C. for a visit, for hanging out for a year and learning some of this stuff, we'd be happy to help you develop proposals to your host countries if you have funding opportunities in your host countries, and to Fulbright to do these kinds of research, to develop these kinds of research tools and opportunities. So thank you all, I appreciate it. And I see there are a number of stuff in the chat. I haven't been very good at looking at the chat, but I'll try and go through it, but also happy to open it up to questions from the audience in general.
Thank you very much, Professor. That was quite perfect. Okay, so I have two questions. They were submitted to the form, but before I start them, you can just raise your hand if you want to ask any question. You can also submit them to the chat box if you want.
So one question here is, Telescope is for metatranscriptomics, while Pathoscope is metagenomics. Did you use any of these tools for inferring function in microbial samples? That's actually, so that's a topic of a grant proposal that has been declined, and we're resubmitting, working on resubmitting it. But it's basically Metascope, right? So we're into the scopes. So we want to enhance Pathoscope 3.0 will effectively be Metascope. So enhancing Pathoscope to have more tools explicitly for doing transcriptome analyses. So we're thinking about it. We have some ideas on how to do that. We have lots of things we'd love to do. We got a good score, but no funding. So in fact, I have a Zoom call with an NSF program officer later today to ask about exactly that topic, funding that topic. So right on, that's where we're heading. We're hoping to get that one funded. We did get a really nice grant, like $1.8 million from National Science. Well, it's been recommended for funding. Hopefully it'll come in here in the next couple of weeks. For expanding our Omics Eye software, specifically for looking at COVID. So that will be huge for our software development team and moving these methods forward. So that's really nice funding. Another question, I'm curious to know what the efficiency in terms of CPU, RAM usage for Pathoscope 2 is in comparison to other benchmark tools. In terms of CPU, it kind of comes down to how you look at these sorts of things, because one step in the Pathoscope analysis is actually developing the reference database, and that takes a long time and uses a lot of RAM, because you put these genomes, your reference genomes, into memory to do the mapping. We've had some issues where, because we use Bowtie 2 has like a 4 gigabyte constraint on how much stuff you can have in RAM. So, and of course our reference databases tend to be much larger than that. So more on the order of 20 gigabytes. So we've had to basically parallelize parts of our analysis, the mapping stage, so we can do Bowtie in 4 gigabyte chunks across our reference database. So creating the database takes time. Once you have the database computed, assembled, then the CPU time for actually doing the analysis is trivial. It goes very quickly, but it can use a lot of RAM. We have in our cluster a couple of high RAM nodes, 120. Well, no, I think we have like 4 terabyte nodes. We have four terabyte nodes. So we purchase those mainly for genome assembly that needs a lot of RAM to put all those pieces of the puzzle together. I don't think we have to go that far. It's mainly because of this Bowtie 2, we're stuck with 4 gigabytes of RAM need, which the nodes accommodate just fine. But we're also talking about part of what we want to do with the Metascope was move away from Bowtie 2 as our mapper. There's some other mappers out there that don't have that memory constraint that we thought we might be able to use as well. So that's about as much as we've done in terms of looking at memory requirements.
I think there is a question. Is there someone who's raising his hand? Darren, can you please speak? Darren?
Yeah.
Oh, sorry. Darren?
Sorry.
Yeah.
Hi, Keith. Thanks for the great talk. Sorry.
So Darren and I know from a long time.
We co-authored a paper a couple of years ago, I think. No, I was wondering whether there's any scope for, so you're talking about, first of all, about all the microbes as being pathogens, which is probably not the case, right?
Yeah.
So is there any scope within the just using read mapping and with the available RNA-seq data, where you've got 10% of the data is the pathogen or the microbe and 90% the host. A lot of the hosts, in inverted commas, are going to be the actual hosts of the microbes that are there. For example, they're going to be viruses and bacteria that infect fungi. There's going to be viruses that infect bacteria and all that sort of thing. And is there any way, and this is a problem that we have with our metagenome studies, of trying to ascribe or associate the virus reads to potential hosts? So potential fungi that might be the host of those viruses, or potential bacteria that might be the host of those viruses, using the data that you get and using your transcription mapping approach, or your transcript mapping approach that you use, particularly considering that about 90%, I assume, of the virus sequences are going to be unseen in the database.
Yeah, that's the biggest issue, right? Is we can only do, well, to a certain extent, we can only do as well as our reference database. And so, to the extent that you have a diversity of organisms represented in your reference database, you can get some reasonably accurate results with that. I want to comment on the pathogen, because this has kind of been an issue with Pathoscope. I mean, obviously by the name, our initial funding on this was actually from the U.S. Department of Homeland Security. And we started working on this back in the day when people were sending envelopes to U.S. Congress people with white powdery substance, right? And people were dropping dead. And Department of Homeland Security was very focused on not just pathogens in general, but agents of bioterrorism. So we actually developed this initially to identify agents of bioterrorism, which is why, hence the name. And that's kind of why we also want to move on to Metascope, because now it is not, we had to do some tweaking because we were hypersensitive about pathogens, but we had to do some tweaking of the parameters to kind of back off that and do better characterization of the microbiome diversity writ large, which it does very well now. So it actually does look at the microbiome characterization writ large, but you have to, it's dependent, it's dependent to a certain extent on the reference database. One of our first examples was there was an E. coli outbreak a few years ago in Germany, and people started dying, and it was from an unknown strain of E. coli that hadn't been sequenced before. When we ran the data through Pathoscope using our reference database of E. coli, we could identify what turned out to be the sister taxon to this new strain of E. coli. 85% of the reads mapped to that sister strain. So you had really good resolution to the closest thing in the reference database, given that the actual organism wasn't in the reference database. But then when we took the sequencing reads, well, the partially assembled genome from this new isolate of E. coli that they did some genome sequencing in this new, and they put the data on GenBank. They had partially assembled, they didn't have a completely assembled genome, but even the partially assembled genome, when we put that information in our reference database, 99% of the reads mapped to that. So we got strain level differentiation. We got the sister taxon when the true thing wasn't there at some reasonably high confidence level. And we got at a super high confidence level, the thing that was actually there when it was there. So one of the things we wanted to do with the Metascope, with our next version of the software, is really develop some statistics around this, right? And some visualization. We have no visualization we'd love to have a visualization so you can actually see where the reads are mapping on reference genomes. Because you can imagine that if you have 200,000 reads mapping to a particular genome, but they're all mapping to one gene, that you have less confidence in the call of that thing in your sample than you do if they're distributed across the entire genome of that reference genome. So we want to we want to develop some visuals and some statistics around testing these kinds of hypotheses. So those are kind of all on the horizon. I think they all come back to your question about, looking at in more detail of what's going on and that organismal diversity that's going on. The other thing is, you also, it turns out bioinformatics isn't the only thing going on here. When we did our asthma study, the first thing our collaborators said was, where are all the viruses? And we thought, oh crap, our algorithm is doing something stupid with respect to viruses. Did we not put those in our reference database? What's going on? So we artificially inserted a bunch of viral sequence reads at kind of a titrated level to see, how low of a representation of viral genomes could you get and still identify virus in those samples. And it turns out you get quite low, and we still identified the virus. So after we went through a bunch of exercises, we went back to them and we said, we've investigated our software, and we're pretty sure that if viruses were in that sample, we would find them. And you expect viruses to be up a kid's nose, right? I mean, that's just where they are. And so we said, how are you guys extracting DNA, RNA? And it turns out the RNA extraction protocol they were using basically eliminated all the virus. So you have to, there's crap that can happen on the wet lab end too, that can screw up your inferences, as well as on the bioinformatics end. So it's important to keep both those in mind and be investigating both ends of the process, right?
Thanks, Keith.
All right.
We have another
Baka.
hand here. John Baka.
Hi, good afternoon, everyone. Thank you so much for the nice talk, Keith. Very informative. I'm interested in the comparison you made between your tool Pathoscope and Chime, especially in 16S. So you showed that you had better results in terms of species assignments than Chime. And did you manage to understand which step of the process you were doing better, or you are doing better than Chime? And have you tried to be interoperable? So meaning producing QZA or QZV files that would enable people to also use the same type of tools we use to visualize results from Chime, but to be able to visualize your results.
Yeah.
I also pasted a question in the chat, just a technical one. Thanks.
The general the general output of Pathoscope and Telescope, for that matter, is a CSV file of in Pathoscope's case, it's a CSV file of number of reads mapped to taxon IDs. So you can take those raw numbers and do any kind of statistical analysis that you want using any of those downstream tools. And in fact, one of the things that we had in our proposal that was not funded was, I saw in the comments somebody asked about why we're programming in Python, and wouldn't it be better to use a workflow DSL or something like that. One of the things we'd actually like to do, my collaborator Evan would like to do, is when you talk to the computer scientists, they think this is a crazy thing to do, but Evan and I are more statisticians, and so the computer scientists want us to use C, C++ to do real software, but the downstream users and want us to use R, right? So what we want to do is basically make an R shiny, so that it's a little more point and clickable to get the main results from Pathoscope, but then also capitalize on all those R tools. I mean, there's a whole human microbiome project tool set, and capitalize on all those downstream tools for analyzing the data. But our basic data output is completely compatible with those sorts of tool sets in R, right? We don't package them together. One of the advantages of Chime is they basically scarfed up every tool you might imagine using and chucked it in Chime. So you can just do everything within one environment. And we're 4 years behind Chime in terms of development, and we don't have near the team that those guys have. But we're also 6 years ahead of Chime in terms of data. I mean, Chime is 16S. And they don't do, in fact, in our grant proposal that was not funded, we actually partnered with Greg Caparicio, who's the principal architect of Chime, and you know, proposed Pathoscope as the Chime metagenomic software that we just, you know, do a Pathoscope plugin for Chime and be done with it, right? So that's where we're heading. That's where we'd love to go. There is, I'm gonna look up this software. There is this software that Evan's group has developed. I'll put it in the chat here, called Animalcules, which is basically our Shiny app for doing a bunch of the downstream analyses. So it is developed by the same, you know, author of Pathoscope. So it definitely takes Pathoscope output directly and then integrates a whole bunch of downstream R tools for doing microbiome characterization. And ultimately what we want to do is link an R version of Pathoscope with Animalcules and bring them together. So you can do basically what Chime does, only for metagenomics and metatranscriptomics and not just 16S data, but do the whole pipeline from data cleaning through end analyses all in one platform.
John has also another question in the chat box. It was about the rhino study. What about it was about the sample R13? Yeah.
Yeah, actually, if we can get back to those slides. And also, there was the question of how comes you outperform Chime, and if you had looked into the reasons about this success.
Yeah, it's principally because the metagenomic data just gives you well, but this is with 16S. I think it's still the reassignment method. It's the treatment of the ambiguous reads, that Chime is still taking ambiguous reads and basically calling higher level taxonomic categories where ambiguous reads are no longer ambiguous, and bidding them in higher level categories, whereas we're using them and making assignments at lower levels and doing that accurately. So it's and we're not alone. Steve Salzberg's group and their Kraken software, they actually, our paper is a master's thesis from a student from Evan's lab. The oral microbiome comparison paper on metagenomics, that is published. It's published in PeerJ, but the 16S comparison with Chime is not yet published. Evan's student is still working on it. But Salzberg's group has done effectively the same study, comparing Kraken to Chime, and Kraken also outperforms Chime at the genus and species level, and presumably for the same reason, because they're also getting better resolution with these ambiguously mapped reads than what Chime does. So I think that's kind of the nuts and bolts of why we're getting differences there.
Thanks.
And I don't know what's going on with this R13 sample. In fact, we did get more rhino funding to go and do more. We actually got 2 based on the initial study. Now we have 2 different rhino studies, one focused in South Africa on the wild populations, and one studied around the world in the captive populations to do more detailed sampling for each population. Eventually we'll bring those together, but we also are getting blood samples so that we can do metabolomics on those samples as well, and do a deeper dive on what's going on specifically with the iron overload disease in the captive rhinos. So again, that sampling unfortunately has been kind of thwarted thus far because of COVID. But my collaborator at the Smithsonian, I think he's gotten permission now to start planning another trip to South Africa. And I think the vets can now get back into the field at Kruger. So hopefully we'll be able to get some more of those samples, both here in the captive populations as well as in South Africa with the wild populations. So if anybody wants to go rhino sampling, if you're from South Africa, let me know. I'll hook you up. Sounds like fun to me. I guess if you have to take blood samples, maybe not as fun as just picking up an animal, but a little more intense.
Actually, it's fun because you can discover an animal like it's a wild animal. It's not just a normal animal, and it's and probably gonna disappear in a few years. So it's good to discover it since it's still here. I had another question here. It was submitted to the form.
Yeah.
Yes, it was about the you talked about the host and the microbiome metatranscriptome interaction. Is there any specific protocol to study this interaction? Specific tool or package to do that?
The functional interaction, you really have to. So there are two ways to do this, right? One is to collect RNA-seq data and make functional inferences from both the host reads and the microbe reads, and start looking at functional predictions. One of the reviews we consistently get on our papers when we do this is that, yeah, but you're only showing associations, so you're not showing mechanism. So, and there are a couple of ways to get to mechanism. One is to do the deeper dive into the pathway analyses, especially if you have other supporting data like metabolomics data, where you can really start looking now more deeply at pathways and what's going on in associated metabolites. The other way is with our cancer studies. One of my collaborators is Kate Chapinelli, who actually has a study system, a cell culture system for ovarian cancer, where we can actually do manipulations where we can express specific transposable elements and see their impact on regulating different genes, different host genes, and look at their impact on those host gene expressions. And we know the host genes do whatever they do in the pathway. So there are a few systems where you have cell lines, cell cultures. Prostate cancer is another one where I've got a colleague who has a mouse system where she has cell culture where you can do where you can overexpress or express transposable elements, endogenous retroviruses, and look at their downstream impact, show that causality, right, relative to what genes are being impacted and what pathways are being impacted. So that's harder, especially for a bioinformatician, right? Because somehow that involves wet lab work, but that's why we like to collaborate with folks who have wet lab capacity and can do those kinds of studies. And so it's all, for us, all about that collaboration.
Thank you. We have another raised hand. Coyote?
Coyote?
Yeah. Coyote?
Take yourself off mute.
Hello, can you hear me?
Yep, you're good now.
It's okay. Thank you, Prof, for the presentation, and I really enjoyed the presentation. And actually, the question I have is that you made mention of a Pathoscope. I actually want to know how this, how it can be used or the relevance of a pathoscope to advance livestock breeding and production? That's my question. To actually improve livestock breeding and production, the use of pathoscope.
Livestock breeding and production. So we've actually, I told you I like to have visitors come to the lab, and I've had a couple of visitors now, a faculty member and a graduate student come from Bangladesh, and both are veterinarians and work on cattle, and they're interested in mastitis and looking at microbes associated with mastitis and figuring out how to improve the animal health and milk production from those cattle. So we've been able to use that explicitly with respect to milk production and cattle through these collaborators in Bangladesh. We also have worked with the poultry industry here in the U.S., butterball turkeys. And the poultry industry gets hit by viruses and bacteria regularly, and their strategy is to, once dead birds start stacking up, they basically kill everything, clean everything, and start over again. And I've suggested a better surveillance strategy is actually to survey for pathogens, for bacterial and viral pathogens, and start identifying some baseline bacterial viral diversity that goes on in your farms, and then identify when you have spikes of pathogens going on. Intervention before you have spikes in dead animals, right? That it's actually surveillance. They don't like it because it's expensive, but hopefully in the future these sequencing expenses will go down. We'll get better sequencing technologies, more portable sequencing technologies, things like MinIONs that we can bring to sites, load up samples, and do screenings of things like pathogens. So I think we're not quite there yet for production. We've done this in human health. I have a biotech company that I started that does this with human samples. But again, it's very expensive, and sequencing costs are always coming down. The technology is very good with low numbers of reads. If you're doing high volume, if you were doing it for surveillance across farms and things like that, I think you could actually get to a pretty good price point, because the trick in all this is just high volume. So your price per sample could go down quite a bit. High volume, low read coverage, and you could really identify when you get lots of reads from pathogens coming out. So I think there's one of our first examples was in aquaculture, looking at fish disease. My student from Chile, in Chile they grow a lot of salmon. There was some outbreak in these Chilean salmon. Fish were dying. It was a syndrome that they hadn't seen before. So pathologically, they didn't quite know how to categorize it. They did PCR tests for all their known bacteria and viruses. Everything came back negative. So they knew. My PhD student had worked in a molecular diagnostics lab before. He was still communicating with his friends in the lab, tell him what he was up to. They said, hey, we've got this outbreak. Can you tell us what's going on? So they sent us dead fish. We ground them up and extracted DNA and sequenced it. And we found both a fish virus and a known bacterial pathogen, a known bacterial fish pathogen that were in these fish in relatively high frequency. And they had the virus they didn't have PCR primers for, and the bacteria they had PCR primers for, but there was actually a mutation in the primer site, so the PCR primers weren't working, even though it was technically a known species. So that's the beauty of the metagenomics, is you can find stuff that but you can find stuff that you don't know, and it doesn't rely on PCR as a window of figuring out what's there and what's not there. So it's a really powerful technology. We've definitely applied it to a variety of agricultural settings, and I think it has a lot of potential in agricultural settings. So thanks. Good question.
Thank you. So there is any other question, please? Just raise your hand or use the chat box.
All right, looks like we're all good.
Yeah.
Jack asked this question. I think this is the last one I haven't addressed. You're doing multiomics data analysis, but I'm surprised to see it's, oh no, I did do that one. Somebody asked about here, Leighton, could you run Pathoscope telescope pretty much on a recent laptop? I think you could, except for the memory requirements. So part of it comes down to the memory requirements for the database. Telescope you probably could because the reference databases aren't that large, but the Pathoscope has quite a large reference database, or at least it can. If you did a custom reference database where you just had, you know, if you have, like if it's, you know, the E. coli outbreak in Germany where you pretty much have an idea what's going on and you just want a bunch of E. coli and Shigella samples that you want to make sure it's, and a few other things that are related, you want to make sure it is in fact E. coli, but you want to get down to strain level and it's a relatively small reference database, you could do that on a laptop. But if you have no idea what's going on and you want general microbiome characterization and you want to use everything from GenBank, then no, that's not going to work on your laptop. You need something that has bigger memory capacity for doing that kind of analysis.
Kyra, you have another question?
Yes, I have. Yes. Actually, the question is very simple, and is directly to the professor of the director of the Bioinformatics Institute, George Washington University. I actually want to know, is there any provision for someone like me to get trained, you know, a kind of a hands-on train in order to advance my level of bioinformatics? That's the question.
There are a couple of ways to think about this. We've been known to travel to host countries to do workshops, right, Abdullah? So Abdullah and I know each other because right before the lockdown in March last year.
1 week before the lockdown.
Yeah, I was doing a workshop, right? So we're happy to travel to host countries to do workshops. We do that regularly. We're also happy to host visitors. If you can figure out how to get here, and there are lots of different mechanisms. So we're very happy to work with you. I've worked with a lot of folks who are interested in Fulbright scholarships for identifying funding opportunities. You know, we can help develop, they usually require some kind of research proposal. So I help you develop the research proposal, provide a support letter. We've been very active in hosting. I've had Fulbright scholars from Madagascar, from South Africa, from Morocco. So we, as well as a wide variety of other countries, Sri Lanka and Bangladesh, and you know, all over the world, really. So, but the third option is to certain countries have granting opportunities specifically for graduate students or faculty to travel to other countries to receive training or to participate in collaborative research. So we're also help, those usually also require some kind of grant proposal. So I'm also very happy to help develop those kinds of grant proposals and those kinds of, you know, help you take advantage of those kinds of opportunities. I have a new postdoc coming in September from South Korea who, you know, emailed me, said, I want to come to your lab and do shrimp genomics. And I said, okay, find some money and come. And I said, you know, you might look into postdoc fellowships from the National Science Foundation of Korea. I think they have such things. So he looked into it, found the research opportunity. We developed a proposal together. He sent it in, it got funded, and now he's coming for 3 years. So those things, they're out there. You have to kind of look in your own country and see what kind of funding mechanisms there are and what opportunities there are. And perhaps even beyond your own country, there are, I don't know, Abdullah, if, I know there's the workshop we did with you was part of the
International Network of Pasteur Institute. It was the International Network of Pasteur Institute.
So it could be that if you're interested in these infectious disease kinds of things, that you know, something like the network of Instituts Pasteur might have postdoctoral fellowships or graduate training opportunities or things like that. So just, you know, do what good bioinformaticians do and capitalize on Google and and see what you can find, and then we're always happy to host people. Washington, D.C. is not a cheap place to come, but it's certainly an exciting place to come, and usually a very comfortable place for international, well, maybe not for the last 4 years, but now for, again, a comfortable place for international scholars to come and visit. Even the last 4 years, we tried to make it comfortable for international people to come and visit. We provide a buffer between what we do as academics and what may be going on in the White House or Congress or whatever. So we can look at that stuff from afar, but not have to worry about it too much. So certainly would love to have folks who are interested contact me, and let's put some proposals in and see if we can get some funding to have you guys come visit, or funding for us to go to visit you and develop workshops and give talks and do collaborative research. Always happy to do that as well.
Thank you, Professor. So is there any other question? I think we're good so far.
All right, very good.
It wasn't heavy, the list is new. Okay, so before I finish, I would just give the floor to Olaiden. If you have anything to say, please.
Yeah, I would like to thank everyone for attending, and I would like to thank Professor Keith Crandall for the great works and talk. And I would want to request that attendees try to subscribe to ASBCB YouTube channel. I put up the link if you scroll up the chat so that you can get notified when we upload the videos. And that's it. And thank you also, Abdullah, for the effort.
Thank you, Olaiden. Professor Keith, thank you very much. We really appreciate your talk. It was so informative. And thank you for all people who attend this webinar, and see you in the next webinar, inshallah. Thank you. Goodbye. Bye.
Bye.
Thank you all. Thanks, Olaiden. Goodbye.
Thanks. Bye.